On behalf of my co-organizers, Jennifer Guerrero, Joanna Joyce, Lisa Cousin, and Dmitry Gavrilovich, I'm pleased to start this meeting and to remind you that we would like you to stay muted and to submit your questions in the first hour by chat. And for a few minutes after Miki finishes speaking, you can stay on and ask questions directly. Without further ado, I'll turn over the introductions to Joanna Joyce.
Thanks a lot, Judy. So it's really, it's a pleasure to be introducing Michaela or Miki de Palma to you all today. I'm sure he needs no introduction. He's a colleague of mine here in Lausanne. He's also a very good friend. Miki's a tenured associate professor at EPFL, the Swiss Federal Institute of Technology here in Lausanne in Switzerland. As you probably all know, and you certainly will hear when he speaks, he's originally from Torino in Italy. He received his master's degree in 1999 and later his PhD in 2004, both from the University of Torino. He studied the anti-proliferative functions of type 1 interferons for his master's degree and the contribution of bone marrow-derived cells to tumor angiogenesis for his PhD. Miki did his postdoc at the Telethon Institute for Gene Therapy in Milan in the lab of Professor Luigi Naldini, who's a pioneer in gene therapy. And there he developed gene transfer strategies for reprogramming monocytes into anti-tumoral immune cells. And excitingly, this work has led to first-in-kind clinical trials of engineered monocytes in patients with brain cancer. That's actually being conducted at the Telethon. Miki joined the School of Life Sciences of EPFL in 2012. And his current research program focuses on mechanisms of tumor resistance to immunotherapies mediated by myeloid cells and the angiogenic vasculature, and a very exciting research direction on the development of engineered dendritic cell vaccines. Miki serves on multiple editorial advisory boards, including Science Translational Medicine and Cell Reports. He's received 2 ERC program grants and was awarded the Robert Wenner Prize for Cancer Research in Switzerland in 2017. So with that, I'm very pleased to hand over the screen to Miki, who will speak about his lab's latest very exciting research on microenvironmental resistance to PDL1-PD1 blockade in lung cancer. And given, of course, the nature of this network, he will naturally be talking about the importance of myeloid cells in this fascinating biology. So over to you, Miki. Thank you.
Hey, thanks, Joanna, and thanks all for this kind invitation. It's a big pleasure to see you all together after a long while. I hope to see you in person more often from now on. So yeah, Joanna, you introduced the topic of my presentation today. I will actually want to discuss a little bit some concepts that relate to the vascular immune interface in cancer and how this can be harnessed to improve the efficacy of immunotherapy. Before that, I just want to mention that I received and currently hold sponsor research grants from Roche, Deciphera Pharmaceuticals, and Avir Therapeutics, and that I sit on the advisory boards of a few companies. So I will start with an overview of the vascular immune network and the work that we have done in the last few years to try to understand how this works in tumors and how this can be therapeutically targeted. So I will start with this very basic and probably oversimplistic cartoon, which nevertheless illustrates the differences between normal blood vessels and tumor blood vessels and how they differentially regulate the trafficking of T cells in tumors. So normal vascular beds are very well structured, as you know. They are virtually permanent in our body. The blood vessels, even tiny blood vessels like capillaries, are fully coated by neural cells that we call pericytes. But even if these vessels are so well structured and quiescent, they are very, very prone to enable the infiltration of immune cells. But this is tightly regulated. For example, when we have inflammation, some mediators can upregulate the expression or induce the clustering of adhesion molecules for T cells. And these enable their extravasation to the site where inflammation is present, for example, a site of infection. Tumor blood vessels are very different. They form through a process that I like to call emergency angiogenesis. And these vessels never mature, never acquire the stable features of normal blood vessels. They are leaky, dysfunctional, poorly covered by pericytes. And paradoxically, these vessels are less permissive to the extravasation of T cells, at least in some contexts, in some tumors, depending on the tumor microenvironment. And what we know is that several tumor-derived factors can actually abrogate the expression of adhesion molecules for T cells. So some tumors, as a consequence of these dysfunctional blood vessels, are poorly infiltrated by T cells. And as you may understand, this is an issue for the application of immunotherapies. My lab has been studying angiopoietin signaling for many years. Angiopoietins are a family of endothelial growth factors or endothelial modulators that bind to the Type II receptor expressed on endothelial cells. And in normal vessels, in normal organs, so in the absence of ongoing angiogenesis, angiopoietin-1 is the main ligand of the Type II receptor, which actually instructs vessels quiescence, so the phenotype that I described earlier. Instead, in tumors, another angiopoietin is highly expressed. This is called angiopoietin-2. It binds to the same receptor, which is called Type II, but actually the outcome of this binding is different. You have destabilization of the vessel, which makes it permissive to sprouting angiogenesis, especially if VEGF is present. So angiopoietin-2 actually works to destabilize the vessel and enable angiogenesis. And my lab and others have shown that you can therapeutically target angiopoietin-2 alone or in combination with VEGF to inhibit angiogenesis in tumors. And this is something that has led to clinical testing more recently. But what we speculated is that in addition to blocking angiogenesis, perhaps you can manipulate the angiopoietin-2 pathway, and so block angiopoietin-2 in particular, to actually reverse the dysfunctional properties of the vessels into a form that makes them more permissive to cell extravasation, so a sort of normalized phenotype, probably by removing the competition with angiopoietin-1. And so this is a concept that we explored in the lab now for many years in many different cancer types. And we have been lucky several years ago to obtain a monoclonal antibody, which we call A2V, and you will hear this acronym frequently in my presentation, which is a monoclonal that is bispecific. So it can block both VEGF-A and angiopoietin-2. So with one molecule, you block the two key pro-angiogenic factors. And so this is possible because the antibody has one arm that is specific for VEGF-A and the other arm that is specific for angiopoietin-2. And we obtained a surrogate version of the clinical antibody, which is called vanocizumab. An antibody that can be used in mice because it's muralized. And one of the models that we have been used in the lab is the polyoma middle T breast cancer model that Jeff, Lisa, and Joanna, and many others have been using now for many years to study the properties of tumor-associated macrophages, because this model is actually in part regulated by the functions of macrophages. And this is an interesting model because these mice develop, owing to expression of a viral oncogene, mammary carcinomas that progress over time, and they are also metastatic. You see lung metastasis in the majority of the mice. So this is a nice model to study tumor evolution in mice. And we performed a number of preclinical studies using the A2V, the anti-Ang2 VEGF antibody in this model. And what we found essentially is that this antibody does the job that it's supposed to do. It's an anti-angiogenic drug. So it can actually prune the majority of the vessels in a tumor, and this leads to substantial tumor necrosis. So the vascular density drops, the tumors are smaller, they grow much less, yet they are more necrotic. So this is an antibody that has substantial anti-tumoral activity in preclinical models of cancer. But as I said before, we were more interested in the immune angle of this process. And so we modified tumors to express OVA, so a model antigen, and we studied antigen-specific immune responses. And what we found, for example, is that A2B alone, so a pure anti-angiogenic drug, can increase in the tumors the abundance of dendritic cells or inflammatory macrophages that actually present the ovapeptide. So there is a sign of an activated immune response that actually translates into better activation of T cells in the tumor microenvironment, as shown here by the higher frequency of interferon gamma positive CD8 T cells. So these are initial signs of a potential immune response that is triggered by blocking VEGF and angiopoietin-2. And actually, I must add something to the vascular phenotype that we observe in these tumors, because not simply the majority of the vessels regress in response to the anti-angiogenic treatment, but what we found is that actually some vessels remain in the tumor microenvironment. And these vessels acquire features of normalized vessels. This effect is largely dependent on blocking angiopoietin-2, not really VEGF. And these vessels really resemble normal vessels, as you see here. So there is a clear conversion of these irregular leaky vessels into stabilized, fortified blood vessels. And this is shown here by the increased pericyte coverage, for example. So, as I said before, normalized vessels are more prone to the regulated extravasation of T cells. And indeed, this is something that we found in our tumors. So we found that there was a better extravasation of CD8 positive T cells, as you see here. In these A2B treated tumors, you have this normalized vessel. You see that they are more regular, more straight than in the control. And you have many more perivascular CD8 T cells. This is the result, in our view, of improved extravasation of the CD8 T cells, which is quantified here. So this is quite interesting and promising because it suggests that a pure anti-angiogenic drug can essentially modulate the immune tumor microenvironment to a form that, if you will, is hotter or at least warmer than the original tumor. So you have increased numbers of immune cells owing to vascular normalization. And so this is something that actually could be exploited for improving immunotherapies. And why I say so? Because, for example, in this study, where we measure antigen-specific, so OVA-specific CD8 positive T cells, we see that in these tumors, the number of OVA-specific T cells is dramatically increased compared to the IgG-controlled tumor. So you have some tumors which contain up to 15% of the CD8 that are OVA-specific. And this is a very substantial response if you consider that we didn't use any immunotherapy here. It's just anti-angiogenic therapy. And of course, this also translates into substantial benefit. Lisa and the others who are familiar with this model know how challenging it is to delay growth of these mammary carcinomas, which develop continuously in the mice, and actually A2B is able to extend survival significantly, while the monotherapies with the just blocking Ang2 or blocking VEGF alone have very minimal effects. So this has really set the stage for now better understanding what was happening at the level of the immune response. And one of the things that we did was to isolate endothelial cells from these A2B treated tumors and to study their phenotype. And what we found is that these endothelial cells actually showed an inflamed phenotype characterized by strong TNF, interferon gamma, and a STAT1 signature, specifically in the endothelial cells. And this was associated with the upregulation of several adhesion molecules, including for monocytes and T cells. But another thing that we found, and this was quite not unexpected, I would say, but quite unwanted, if I may, is that PDL1, so the ligand for PD1, was also significantly upregulated in the endothelial cells, both in the RNA-seq study and by flow cytometry. So there is increased expression of PDL1, which of course could potentially limit the activation of T cells in the tumor microenvironment. And actually, we found that this effect was probably largely due to the improved extravasation and trafficking of the CD8 T cells, because whenever we have accumulation of perivascular CD8 T cells, which, as I shown before, are often activated, so express interferon gamma, we see upregulation of vascular PD-L1. And this effect was actually interferon gamma dependent, because in experiments where we blocked interferon gamma, we saw that this upregulation was abrogated. So this was a sort of, unwanted effect, and actually A2B was on the one hand promoting activation of the immune response by increasing antigen presentation, by increasing trafficking of T cells, but on the other hand was also inducing PDL1, probably as a feedback mechanism, owing to the greater amounts of CD8 T cells that are now activated and surround the vessels. So the most obvious thing to do was to explore the benefits now of blocking the PDL1-PD1 pathway. And this is actually what we did. So we used, in this case, an anti-PD1 antibody, but we also did experiments by blocking PDL1. And what we found is that we can provide additional benefits in breast cancer, melanoma, some models of colorectal cancer. This is a gated transgenic polyoma middle T model where you clearly see that there is a fraction of the mice who live longer. It's about 30% in the specific experiment. Is not all of the mice. Actually, we don't understand whether we have antigen-specific responses in this model. It's been very difficult to identify antigens here. But what I can say is that this response is T-cell dependent, because when we deplete the T cells, we lose this benefit. So clearly, there are benefits of co-targeting, of reprogramming the tumor blood vessels into a normalized form and adding immune checkpoint blockade. But as I will show in a minute, not all cancer types benefit from this type of therapy. And in the course of the studies, we also collaborated with a couple of groups to explore alternative immunotherapies. And these are agonistic CD40 antibodies. I'm sure most of you are familiar with them. Essentially, CD40 is a co-stimulatory molecule that is expressed in part on antigen-presenting cells and macrophages, and this is required for the full activation of antigen-presenting cells. And a number of agonistic CD40 antibodies have been developed that can actually energize dendritic cells. These have been tested now also in clinical trials with I would say, promising results. So this is a collaboration that we did with Alfred Zipelius lab in Basel, where we studied combinations of A2B with CD40 agonistic antibody. This is one of the experiments that we did, where we use a transplantable tumor model, MC38. And what we found essentially was that the monotherapies, as expected, could maybe delay the growth of some of the tumors, but were poorly effective. A2B was better effective at delaying tumor growth, but the combination of A2B with a CD40 agonist antibody actually was very effective. Here we have the majority of the tumors which fully regressed at the beginning, and the few that could grow showed a delayed response, even if a delayed growth, even if when growth happens, this can often go fast. So this was encouraging, and actually what we found are similar findings compared to what I've shown you before with the PD1 antibody in combination with A2B. So we normalize blood vessels, we increase the trafficking of CD8 T cells to the tumor microenvironment. So the mechanism is, at least in part, similar, with the difference that here what we noted are major effects on tumor-associated macrophages. So something that you will see also in my next slides is that A2B alone decreases the abundance of F480 positive macrophages in the tumor microenvironment. And we believe this is one of the reasons why it has anti-tumoral activity. But what you can see here, that the combination of A2B with CD40 agonism decreases macrophages even further. And in particular, what it does is to shift their phenotype into a sort of more immunostimulatory phenotype. This was assessed by a number of typical markers, but we also performed RNA sequencing analysis of several cell types in the tumor microenvironment, including endothelial cells, T cells, and macrophages. And what you can see here is that when you combine A2B with CD40, there is a very strong activation of let's say, STAT1 dependent pathways in the macrophages, so interferon gamma signaling, TNF, IL-1, and toll-like receptors. So these are all signs of an inflammatory, immunostimulatory activation of the macrophages that very likely contributed to the effects that I've shown before. And then we also collaborated with Tanya Petrova at the UNIL. Actually, this is work from her lab to which we contributed. Tanya developed colorectal cancer models that are transgenic. Essentially, here I will show a bit of data involving two transgenic mouse models of colorectal cancer. One is what we call AP, that is an APC p53 deleted mouse model, which is induced by intrathecal tamoxifen injection. And then there is another mouse model which she developed that also involves the deletion of PROX1. And the difference between these two models, the AP and APP, is that the APP model, in which PROX1 is deleted, actually gives rise to colorectal cancers in mice that resemble the desmoplastic, highly angiogenic, and chemorefractory colorectal cancers also found in humans. So this is a very, very nice model of colorectal cancer that we know is resistant to the majority of the therapies. And one of the features of these desmoplastic colorectal cancers is that they are virtually devoid of T cells, including CD8 T cells in the lamina propria, so in the tissue that surrounds the tumor. So there are very scant T cells, and these make them probably less prone to immunotherapies. And as you can see here, these tumors are also chemorefractory. In fact, when you treat the AP tumors that are not desmoplastic, you have a response to 5-FU, but instead when you treat the desmoplastic tumors by 5-FU, you have a paradoxical effect, which is actually also seen in patients with this subtype of colorectal cancer. So the tumors seem to progress even faster. So this is a very intractable tumor. But the good news is that a combination of A2V and CD40 agonism actually could control these tumors. There was a statistically significant reduction. And actually, these also translate in widespread destruction of the tumor stroma. So the desmoplastic tumor stroma is reduced. There are large necrotic areas in the epithelial compartment. So this combination of A2B with the CD40 agonist was very effective in this model. And actually, these tumors become infiltrated by CD8 positive T cells, which are not anymore limited to the stromal compartment of the tumor, but now can also infiltrate the epithelial compartment and destroy the tumors. So what I would like to add is that in this model, there was no efficacy of combining A2V with PD1 blockade. And I will elaborate on this in the next part of my talk. So what I've showed you so now is the opportunities of combining anti-angiogenic agents. In particular, we believe agents that also involve the use of Ang2 blockade with immunotherapy. And essentially now this concept, our group published this study in Science Translational Medicine, along with a very, very nice paper from Gabriele Bergers, who also came to similar results and also identified the formation of high endothelial venues as a major determinant of a good response to anti-angiogenic immunotherapy. But essentially now a multitude of studies have shown the benefits of this approach, whereby anti-angiogenesis reprograms the tumor blood vessels, facilitates T cell trafficking, and then immunotherapy can actually enhance the efficacy of the T cells. And this has also come to clinical realization. For example, this is a New England Journal of Medicine study published last year showing the benefits of combining PDL1 blockade with anti-VEGF blockade in unresectable hepatocellular carcinoma. And this actually provided much better benefits compared to standard of care, sorafenib. And actually the FDA has approved now this treatment as a first-line treatment for unresectable hepatocellular carcinoma. So there are clearly benefits of further exploring this type of combination. But in the second part of my talk, I'd like to discuss a little bit about immune checkpoint blockade and potential combination with anti-angiogenics in lung cancer. So as you know, PD1 or PDL1 blockade is now approved for the treatment of non-small cell lung cancer, either in first or second line treatment, depending on the tumor types and the characteristics of the tumor. I would say that, you know, patients experience in a substantial and a sizable fraction of the cases some responses, but the rate of long-lasting responding is dismal. So immune checkpoint blockade is not so effective in lung cancer as we were hoping for. So we have been studying lung cancer in the lab now for many years, even if we haven't published yet about it. You know, it's a long-term effort in the lab. One of the models that we use in the lab is what I call the Tyler-Jacks model. Essentially, here we do intratracheal injection of a tree that activates a mutant RAS, G12D oncogene and deletes P53. And this is a very nice model because you can titer your vector to have a relatively low number of tumors that develop in the lungs of the mice, and these tumors are representative of human non-small cell lung cancer. So they have histological and behavioral features very similar to human lung adenocarcinoma. So we tested our anti-angiogenic immunotherapy strategy. This is one of the very first experiments we did where we compared B20, which blocks VEGF alone, where we used angiopoietin-2 blockade alone, or A2V. And actually what we do in this model is to perform longitudinal weekly tumor imaging by micro-CT. So we define individual tumors and we monitor how individual tumors develop over time. Essentially everything that is above this dotted line means progression. Everything that is below means regression. As you can see here, even if these treatments, VEGF alone and A2B even more, induce some tumor control, there is no bona fide regression of the tumor. So we see tumor stasis or substantial control with A2B. So we said, well, probably we can enhance now this effect and achieve regression by combining with checkpoint inhibitors. But here we found very, very interesting and in my view, provocative data. So actually, when we combined A2B with the A2B plus PD1, we saw that a fraction of the tumors actually progressed faster than when we gave A2B alone. And I can say that we saw similar things when we combined cisplatin with anti-PD1. In some cases, we saw this acceleration. So there was a nice progression under anti-angiogenic therapy. Of course, this was a major blow initially to the project, but as it happens, we got some interesting insight from here on. The first thing that we asked is whether these tumors are poorly immunogenic so that they don't benefit from this combination. And so what we did is to further engineer these mice to delete a mismatch repair enzyme, MSH2, which by the way is inactivated in a fraction of non-small cell lung cancers. And so when you delete MSH2, you're expected to increase the mutational load into these tumors that develop over several weeks. And actually, when we analyzed these tumors, we found that they carried higher numbers of non-synonymous mutations. And this was very interesting. Actually, they had greatly increased, at least in a fraction of the tumors, not in all the tumors, but a greatly increased number of immune cells and more specifically T cells. I must say that these tumors are histologically very heterogeneous, like human diseases. So you can see, different phenotypes, but overall, this increase in T cells was quite noticeable and clear. So we combined A2B with PD1 also in this model, which is expected to be potentially more immunogenic, but still no benefit. Actually, the tumor seems to grow a bit faster. And we did additional studies where we also introduced a nova peptide into these tumors, but we obtained similar results, no benefits of the combination. So that was pretty disruptive at the time. So we also said, well, maybe these models, these transgenic models have inherent variability. We will never get phenotypes, clear phenotypes. So we also did a clonal, isolated a clonal population of KP cells to have tumors that are standardized, homogenized among mice. We did the treatment, and again, the combination of A2B plus PD1 was worse than A2B alone. So this was a clear effect that was reproducible across different tumor models. So we started to study these mice. And what we found is that vascular normalization that is induced by A2B actually, in this model, disproportionately increased the number of tumor-infiltrating Tregs. So there are already quite high baseline levels of Tregs in this model, but A2B was actually increasing them. They go hand in hand with the CD8. The CD8 increased, but also the Tregs increased. And this was quite you know, it suggested that probably the Tregs were involved in this process. So we studied the Tregs, and actually we found that when we compare Tregs with the other CD4 positive cells, Th1 or CD8 T cells, actually the Tregs expressed much higher levels of PD1 in these tumors. They are PD1 high. The data are quantified here, where you see that actually between 60, 80, 90% even of the Tregs are PD1 positive in these tumors. While if you look at the CD4 positive non-Treg cells or the CD8 positive cells, the fraction of PD1 positive cells is clearly lower. So in these tumors, there is clearly higher representation of PD1 positive cells within the Treg compartment, and the expression level is also higher. So we decided to have a look at the site where our PD1 antibody was actually going in the tumor microenvironment. And we found that actually the PD1 antibody was decorating very efficiently the FOXP3 positive Tregs, but much less so the CD8 positive T cells. And this, if you will, is expected because the FOXP3 Tregs express higher levels of PD1 than the CD8 positive cells. But nevertheless, this was a very, very provocative finding, because essentially now what is happening is that when we administer the PD1 antibody, this is actually mostly binding to the Tregs. And what we found in the tumor microenvironment of these tumors that are treated with either PD1 alone or in combination with A2V is that, for example, IL-10 was upregulated. You will often see in my slides quite some variation, but this is intrinsic to this transgenic model. But you can clearly see that on average, these tumors express higher levels of IL-10. And IL-10 is actually indicative of an immunosuppressive microenvironment. The source could be either macrophages or the Tregs themselves. But this could potentially indicate that Tregs get more activated by the PD1 binding antibody. And another observation that we made is that actually the Tregs were frequently in clusters with macrophages. As you can see here, these are tumors that have been analyzed after 4 weeks of treatment, treated with A2V-PD1. You can see these clusters of FOXP3 Tregs that are surrounded by F480 positive macrophages. When you look at the CD8 T cells, which are fewer after A2V-PD1, as I said before, we don't see these interactions. So there are interesting clusters that actually may suggest that there is a crosstalk between macrophages and Tregs. And this is quantified here. The proportion of Tregs that are in contact, direct contact with macrophages, is much higher for Tregs than for CD8 T cells. So this led us to think that probably macrophages and Tregs that are bound by the PD1 antibody actually establish cell-to-cell contacts that support an immunosuppressive microenvironment. In tumors, actually, you all know macrophages are a major source of Treg supportive cytokines, including IL-10, CCL17, which actually may support the expansion of Tregs in the tumor microenvironment. So we decided to knock out macrophages from these tumors. And you know well that one of the best ways to deplete macrophages from tumors is by blocking CSF1R. CSF1R is involved in the differentiation and maturation of macrophages. And we were lucky enough to collaborate with the Carol Arias from Roche, who provided us with an excellent CSF1R antibody, which actually depletes macrophages very efficiently from these transgenic lung tumors. You can see here that some macrophages actually remain in the tumor microenvironment, but they are much fewer than at baseline. So we had a tool now to ask whether macrophages can affect the Tregs, and the answer is yes. So when we concomitantly blocked CSF1R with A2V-PD1, we could somehow, at least to some extent, reduce the frequency of Tregs in these tumors that eventually return to baseline. There is an increase with A2V, further enhanced by PD1 or the combination of A2V and PD1, and they return to baseline levels when macrophages are depleted, suggesting that Tregs are dependent on macrophages. And actually, that was an experiment done at very short term, so it was an acute elimination of macrophages, but we can chronically deplete macrophages using the CSF1R antibody in tumors. So this is a 4-week treatment trial. And here you can see a more, I would say, extreme phenotypes. So these are macrophages in the tumors. They increase with A2B/PD1, but they drop when we deplete them with the CSF1R, not completely, and I will explain why in a minute. Still, they decrease. And this actually controls the Tregs, which are induced by A2B/PD1 in numbers and drop to baseline or even lower than baseline levels when you deplete the macrophages. And in parallel to this, we also saw a drop in important Treg cytokines, for example, CCL17, which is required for the recruitment of Tregs. It's not required, but it contributes to the recruitment of Tregs in the tumor microenvironment. The levels of CCL17 were decreased, as well as the enhanced levels of IL-10 seen after the combination return to baseline level. So there is a clear effect of blocking macrophages on the Tregs. So the obvious question is, does this translate into tumor control? Is this an effective treatment? And I would say yes and no. Yes, in part because you see now the tumors progress much less. This is tumor growth, again, change from baseline. Everything above 0 means progression, everything below means progression. So some tumors now regress. There is clearly a better benefit of compared to A2B/PD1. Consider this is the IgG control, so the tumors progress very fast. A2B/PD1 is effective to some extent, but you should remember, it's not shown here, that A2B alone would be here. So you lose the benefit of A2B by A2B/PD1. And then you go back, you restore partial sensitivity. But eventually, you know, if you look at the mean progression, this is about 50%. Plus 50%. So these tumors on average are still progressive. So how can we increase the response rate? So one obvious thing to do was to include in our regimen cisplatin. Why? Because cisplatin is a drug, is a chemotherapeutic that is also indicated for non-small cell lung cancer, is used in the clinic. But what we found when we combined CSF1R blockade with cisplatin is something very interesting, because we found that cisplatin actually was depleting a population of macrophages which were resistant to CSF1R blockade. So here is a sort of representation of different types of myeloid cells of the mononuclear lineage that we see in these tumors, monocytes, what we call monocyte-derived macrophages that are in orange, alveolar macrophages that are inside the tumor. So there is a population of alveolar macrophages which actually integrate in the tumor, that are in red here. And these are, you know, are essentially the ratios that you see. Then we have other subsets. So now follow me in what happens when we administer the various drugs. So A2B/PD1 doesn't really much change the ratio between alveolar macrophages, monocyte-derived macrophages. When we use CSF1R, actually the monocyte-derived macrophages are wiped out. They completely disappear. The alveolar macrophages are not affected, but they are affected by cisplatin. So they are sensitive to cisplatin. They are eliminated by cisplatin. And the combination of cisplatin and CSF1R virtually removes all mature macrophages from the tumor microenvironment. This is a quantification by flow of the data here. So you can see here, if you look at the alveolar macrophages, they are depleted exhaustively by cisplatin, with or without CSF1R. Instead, CSF1R depletes, at least partly to some extent, the monocyte-derived macrophages, but they are not affected by cisplatin. So combined cisplatin and CSF1R blockade eliminates terms of both origin. And actually, if you now quantify all macrophages, independent of their origin, subtype, in the tumor microenvironment, you can clearly see that you have partial inhibition, partial depletion with CSF1R, moderate overall depletion by cisplatin because the alveolar macrophages are just a fraction. But when you combine the two, macrophages now go down very, very, very efficiently. And how to explain this? Well, a simple explanation is that the monocyte-derived macrophages actually express CSF1R. These are RNA-seq on sorted terms. I don't have time to show you all the data of sequencing that we did because it's too complex, but essentially CSF1R is more highly expressed in the monocyte-derived macrophages than in the alveolar macrophages. Why? The alveolar macrophages express Ki67, and we found that they also express many more markers of proliferation. So we believe that in the presence of tumor, they are proliferative. There is in situ proliferation of alveolar macrophages, which contribute to the immunosuppressive microenvironment of the tumors. Why? Because when we now take the whole tumor and we measure some Treg markers or cytokines like FOXP3, for example, this is shown here. FOXP3 is really abated by the combination of cisplatin and CSF1R together with the standard of care. CCL17 levels drop dramatically. So this suggests that both alveolar and bone marrow-derived macrophages contribute to produce CCL17. And the same is true, at least in part, for IL-10 and certainly for TGF-beta, which is another major immunosuppressive factor in the tumor microenvironment. We also had a look at human data sets. This is a simple analysis from the TCGA where we actually found that a thumb signature that we validated previously in a published study in collaboration with Daniel Spicer actually is associated with genes indicative of Tregs like FOXP3, CCL17. So there is a IL-10, TGF-beta. So there is a correlation between macrophages and Tregs. Again, in support of the notion that macrophages support Treg expansion in the tumor microenvironment of the lung. And now, of course, the efficacy data in this model, and now the data look very good. This is an intractable mouse model of cancer. So this model is completely resistant to checkpoint blockade, as you can see here, is resistant to many chemotherapies, is a very untractable tumor model. And now we can obtain more than 70% regressions and even complete regression. So we had tumors that now completely disappeared when we combine the anti-angiogenic immunotherapy with cisplatin and CSF1R blockade, which I call TAM elimination. It's a cocktail that might also in part affect the cancer cells directly, but we believe that the main effect is on the macrophages. So we have additional data that I don't have time to show, but I would like just to show this cartoon, which may summarize what I've shown today. So macrophages produce immunosuppressive factors that expand the Tregs in the lung. This is not seen in other cancer models, but it's seen in the colorectal cancer model that Tanya Petrova developed in her lab, where PD-1 blockade actually didn't work in combination with anti-angiogenic therapy. So now these Tregs expand, they express high levels of PD-1, they are bound by the antibody, and actually they compete with CD8 for binding of the antibody. And of course, they are suppressive, and this may actually prevent the efficacy of PD-1 blockade. When we eliminate macrophages of two different subtypes with CSF1R blockade and cisplatin, actually the Tregs drop in number because they lack macrophage support. Now, the PD-1 antibody, and this is a piece of data that I didn't show you, preferentially binds to the CD8, which we believe behave as effector functions in controlling tumor growth, because we did experiments where we depleted the CD8, and at least at the early stages, we see a clear effect on inhibiting cancer cells. So clearly, these data suggest that there is a vicious macrophage Treg crosstalk. And recent data that are published from clinical trials also indicate that hyperprogressive disease happens in patients with lung cancer treated with PD-1, PDL1 antibodies. So this is something that is seen in patients. It's still quite a controversial topic, how to define a hyperprogressive disease, but this is what we clearly saw in mice. And we found that by targeting the macrophage Treg axis, this can be overcome. So to summarize about prospects of anti-angiogenic immunotherapy, I've shown you that in some models like breast cancer, we actually do see benefits of combining A2V with PD-1 because these tumors are not enriched in Tregs, but other cancer types, including colorectal cancer and lung cancer, are actually enriched in Tregs. And this actually is detrimental because they would bind to the PD-1 antibody and actually in the presence of macrophages support a vicious immunosuppressive network that eventually would limit the efficacy of of immunotherapy. But as I've shown, we have a clinically viable strategy now to overcome this effect by targeting the macrophages. So these are the people who actually performed this work. I'm really thankful to them. Excellent PhD students and postdoc that over the years studied the polyoma middle T breast cancer, the lung cancer model, and I'm very thankful to them, and I'm happy that some of them already moved on to very good jobs. So I would just take the last few minutes before letting you go. I may realize that you have had enough of me, but I want to show you just a few slides because one recurrent topic is whether we should deplete or reprogram the macrophages. And actually, Joanna has done excellent work in this by showing that in some models, actually CSF1R inhibition is not depleting, reprograms the macrophages. But this is still, an interesting topic, and there are still many things that we need to understand. We have several drugs now that can potentially target the macrophages. As I said, CSF1R, we also have CCL2, CCR2 blocking reagents, but the sobering reality is that in clinical trials, most of these agents have failed, and also in combination with the chemotherapy. So there is still an ongoing interest in trying to understand how we can best harness macrophages. And I just want to take the last couple of minutes to show something that we have done and that we are still doing in the lab in trying to understand the mode and the effects of reprogramming macrophages. This is work that we published a few years ago, showing that actually you can reprogram macrophages to a form that is immunostimulatory, that attracts T cells by deleting dicer. Dicer is an enzyme that actually controls the production of and maturation of microRNAs. And in this study, we found that actually one family of microRNAs was specifically affected by dicer deletion in the macrophages, and it's the LED7 family member. Actually, we found that when we block specifically LED7, we phenocopy the effect of dicer deletion. And when we rescue the activity of LED7 in dicer deficient macrophages, we rescue the conventional immunosuppressive or M2-like, if you want to use this nomenclature, phenotype of the macrophages. But nevertheless, by interfering with microRNA activity that controls a number of downstream pathways that impinge on STAT1 signaling, we can make the macrophages activatory toward T cells. So we did also some experiments in more challenging model like this orthotopic KRAS P53 lung cancer model. And actually we found that when we inoculate these tumors in the lung of these mice that have macrophages where dicer has been inactivated, we have both lower incidence and progression of these tumors. This is a very dramatic phenotype if you consider that the deletion of dicer is really cell autonomous in the macrophages. So there is no, the system is not leaky. So it's just the macrophages that are reprogrammed, not even depleted. They are in equal numbers, and this is sufficient to limit tumor growth. We also moved on to do single cell RNA sequencing analysis. I don't have time to go much into the details of this, but actually we found important reprogramming of the tumor microenvironment where dicer is deleted in the macrophages and the macrophages are reprogrammed. This is very interesting. I don't have time to go through this, but actually we performed trajectory inference analysis, and we found that the myeloid and specifically the macrophage compartment is altered here. And we have a shift toward immunostimulatory macrophages rather than toward immunosuppressive macrophages. But we also saw changes in the T and NK compartment. This is quantification of the differential abundance of all the populations that we identified. Some are grouped together for simplicity. But if you look at the numbers of monomacrophages in the wild type and dicer knockout, there is no change. So macrophages are there in normal numbers, yet there is this broad reprogramming of the tumor microenvironment. CD8 T cells increase, granzyme high NK cells increase. And when we performed, when we analyzed gene sets that are regulated, we see a broad response across different cell types, including the cancer cells, by reprogramming macrophages. You see that the first hits here are interferon gamma response, interferon alpha response, allograft rejection. So there is clear evidence that by specifically reprogramming macrophages, you can achieve remodulation of the tumor microenvironment. And of course, this is well known to many of you. Actually, Judy and many others have shown this, Jan, by different approaches. The key now is to identify viable drugs that can be used to actually enable this program. And I will finish with this slide, where actually it's a simple experiment with a transplant tumor model, but you can clearly see how these reprogrammed macrophages can enable immunotherapy. These are controlled tumors. What you see here is just the growth of tumors in this model, where they are grown in mice with macrophages that lack dicer. So there is tumor inhibition, but not block of tumor growth. And the same is true when you apply CD40 agonism or anti-PD1 antibodies. There is some growth delay. But when you perform this experiment in the dicer knockout context, both CD40 agonism and PD1 blockade can actually sterilize the tumors. So there is a major synergy here by combining immunotherapy in a context where macrophages are reprogrammed. And of course, if you think that, many drugs may not effectively reprogram macrophages, this may explain why several clinical trials, also in combination with checkpoint inhibitors and macrophage depleting agents, didn't work, because we need to achieve the right reprogramming of the macrophages. And I want to thank the people in the lab who again do an excellent job working on macrophage reprogramming we have shown here. So with this, I just go to my acknowledgements. I want to thank my dear colleague, Tanya Petrova, and Alfred Zipelius, Carol Arias, Nadine Zanger, who actually is our bioinformatician, Daniel Spicer and Damia, who actually collaborated to the work that I've shown today. I also want to mention other members of the lab who contributed to the studies that I've shown. But a special thank goes also to the other lab folks who do other work in the lab, which I haven't shown today. We have two main areas of research in the lab, dendritic cell vaccines, extracellular vesicles, and I want to thank them because, of course, through lab meetings and the continuous interactions, they also contribute to what I've shown today. So with this, I'm done. I just want to highlight our founders, and I want to thank you for your patience during my talk.